Welcome to Kingdom Animalia. This kingdom contains the very last of our taxonomy for the semester. For more detailed step-by-step instructions, see the Help with Narrated PowerPoints file in the Cloud Drive. This is a joint product we have put together over the course of several years. We work together in an effort to bring you, the student, the best possible learning experience. We are all firm believers in two heads, or in this case multiple heads, are better than one, and working together makes for a more well-rounded curriculum. We all teach at multiple campuses, so you may see material from more than one source or hear references of other campuses such as Northlake College, Collin College, Angelina College, Grayson College, etc. We do our best to update these slides as soon as possible when new updates come, but updates to the textbook will always trump what is in the PowerPoints. Announcements in class or on Blackboard or Canvas also trump material in the slides. If we find an error, we will let you know. If you spot an error in the material, let us know. We are constantly revising it, and you are often our best editors. Send us a friendly email and we will investigate the issue. If it's a typo and punctuation, etc., we may not get on it immediately, but if it's an error in content, we will definitely address it. After this presentation, you should be able to understand what an animal is, identify the steps in gastrulation, determine the differences between protostomes and deuterostomes, and identify the major clades and the molecular hypothesis dividing animals. Here's a look at the taxonomy of Animalia down to the phylum level. So we are in supergroup Uniconita, major clade Episticons, kingdom Animalia. So what is an animal? This should be a review to you. Nothing in here should shock you. Animals are multicellular and eukaryotic. They do ingestive heterotrophic nutrition, and unlike fungi, they lack cell walls. Animals have highly differentiated tissues, including nervous and muscle tissue. Their reproduction is typically sexual, and they have a dominant diploid life cycle. There is probably an exception to everything, however, this is a really tight group. The origin of animals is suggested to be between 1 billion to 542 million years ago. Molecular suggests the older, fossils support the earlier, probably because it took a while for animals to evolve hard parts that would actually fossilize. The Cambrian explosion is a huge age of animal diversity. It may have been caused by one of three issues: the predator-prey hypothesis that diversity through natural selection, it's basically an arms race. As one organism becomes bigger and badder and larger, other organisms have to evolve in response. There's also the oxygen hypothesis. The more oxygen there is in the atmosphere, the more energy there is available and the more open niches that would have been exploited by those who are able to utilize oxygen for energy. And then the Hox genes hypothesis. Remember, those are the ones that tell us about where things go. So these genes might have evolved during that time period, and as mutations occurred in them, organisms started to change drastically in their traits. The truth is, it's probably all three of these happening together that caused this great explosion of diversity. The Mesozoic era brought about some massive animals like dinosaurs and even the first mammals. Then the Cenozoic era started off with a bang, meaning that this is where that asteroid took out the dinosaurs when it hit Earth. And as the climate cools, grassland apes began to evolve into Homo sapiens. Now let's talk about how we distinguish between animals to classify them. One way we do that is through body symmetry. Asymmetrical animals have no body symmetry, no regular plan of development. There is no way to cut them in half and get an equal right and left half. Radially symmetrical organisms are shaped like a cylinder or a soup can, so no matter how you slice them, you get an equal right and left side. Bilateral organisms is what we are. The organism has a right side and a left side, a dorsal and a ventral, and an anterior and a posterior. So there's only one way to slice them and get an equal right and left half. Next, let's talk about some body locations. We will use these a lot in the next few chapters when talking about organism traits. The first is dorsal, which is the top side, and ventral is the bottom side of the organism. You can see these here. Here's another look at dorsal versus ventral. Anterior is the front of an organism, usually with a mouth, while posterior is the back of the organism. Here's another example of anterior versus posterior. And one more look at anterior versus posterior. Cephalization is all of the sensory equipment located at a concentrated area known as the anterior part of the organism, or often the head of the organism. You can see cephalization here in this flatworm in the region indicated by the red circle. Another way we classify organisms is based on their embryology and how the embryo develops. Fertilization takes place between a swimming sperm and a non-motile egg. Then you have a series of mitosis without growth known as cleavage. This forms a blastula, which in most animals is a hollow ball of cells. Gastrulation follows, which is the process by which layers form, and this starts the differentiation process for the adult body parts. There are two main types of embryo development, protostome and deuterostome. Protostomes have spiral cleavage, which means the cells, as they divide, they are not all the same size. Some are smaller and others remain larger. This cleavage is also determinate, meaning each cell begins to specialize and will only give rise to cells of a specific tissue type. If you remove a cell from a protostome embryo at the 8-cell stage, the embryo that develops will be missing all of the tissues that the missing cell would have developed into. In protostomes, the blastopore, which is the first opening formed in a cell, becomes the mouth. This is where their name comes from, proto meaning first, stome meaning mouth. So the mouth develops first and the anus develops second. Examples of protostomes would be annelids, arthropods, and mollusks. Deuterostomes have radial cleavage, meaning all the cells stay the same size as they divide. This cleavage is indeterminate, meaning the cells do not specialize, so any cell can become any type of tissue. If you remove a cell from an early stage embryo, other cells will form the tissue, so the embryo can still develop completely. In deuterostomes, the blastopore becomes the anus, which is where the name comes from, deutero meaning second, stome meaning mouth. So the mouth develops second and the anus develops first. Examples of deuterostomes are echinodermata and chordata. Here's another look at protostome versus deuterostome development. There are two types of tissue development in embryos in animals. Diploblastic organisms develop from two layers of embryonic tissue. The two layers are the ectoderm, which forms the outside of the organism, the surface, so the skin, to the nervous tissue. The second layer is the endoderm, which forms the internal tissues like the digestive and internal organs. Triploblastic organisms' bodies develop from three layers of embryonic tissue. They have an endoderm, an ectoderm, and a third layer called a mesoderm, which forms muscular tissue. Within the tissue layers, some organisms have body cavities known as a coelom. A coelom is a body cavity that is fluid-filled, separating the digestive tract from the outer body. We classify organisms based on the lack or type of body cavity. Acoelomates lack a body cavity completely. All of their tissues come into direct contact with one another. Pseudocoelomates have a coelom, but it is formed between the mesoderm and the endoderm layers. True coelomates possess a true coelom, meaning it lies in the middle of the mesoderm tissue. Once embryo development finishes, there are two types of development in animals, going from the young organism to the more mature adult. In direct development, organisms develop like humans, where the offspring are miniature versions of the adult, and as they develop, they get larger and eventually reproductively mature. You can see this in the fish life cycle pictured here. Metamorphosis, or indirect development, is sort of the opposite. Offspring are sexually immature like in direct development, but they also look significantly different than the adult form. Think of a tadpole that becomes a frog, or a caterpillar that becomes a butterfly. The larval stages have no resemblance to the adult. In the next few chapters, we will look at the molecular hypothesis for animal classification. We will start with porifera, which are asymmetrical and lack true tissues, move on to radiata, which are radially symmetrical and have true tissues, and then we will finish off in the clade bilatera, which has bilateral symmetry and are deuterostomes and protostomes. There are other methods of classifying animals which you may come across in your research, but there are some areas that all taxonomists agree with. These include all animals share a common ancestor. Sponges are the basal animals and are the clade Parazoa. The clade Eumetazoa is all the organisms with true tissues, so everybody but sponges. Clade Bilatera is generally agreed upon, and clade Deuterostoma, which contains vertebrates and echinodermata. Here's a practice chart for you to fill in and study.
Please consider the following.
Here's how you contact the Hawaii program. You can check out any of these resources. The Twitter feed is excellent for looking at some of the activities.